Okay, the next section we're looking at is nucleic acids, yeah, and looking at the DNA to transcription to translation part of it, as well as some mutagenesis, and also looking at different types of genetic recombination or transformation conjugation of transferring DNA from one cell to the next. But before we get into that, we have to get to the basic background of DNA, right? So we got to look at nucleotides, purines, pyrimidines, what those are, genes, alleles, mutations, looking at basic DNA replication, transcription, and origin of replication of prokaryotic cells, the replication fork, leading and lagging strands, DNA polymerase, ligase, Okazaki fragments, mRNA, rRNA, and of course tRNA. All of these things leading into the transcription part of it. We'll definitely get into translation, but we'll have to break these up into parts because there's a lot of little details, especially if you haven't done any DNA parts in your actual background or other biology courses that you've taken. Okay, for those of you who have taken the biology courses, a lot of this will be review, but make sure you put it into context of prokaryotic organisms and definitely not eukaryotic ones. Okay, so nucleic acids, right? These is a molecule consisting of many nucleotides, which in turn form a polynucleotide chain as a nucleic acid, right? So if you take nucleotides and you string them all together, you'll get a nucleic acid. Nucleotides, oftentimes people think of this as just the base, right, like adenine, cytosine, guanine, thymine, or uracil, but also consists of that pentose sugar and that phosphate, which make up the whole entire nucleotide. So right here you see the actual nucleotide, right, and just making sure here's your nitrogenous base, but don't forget about your sugar and phosphate molecules that make up the backbone of the whole entire nucleic acid. So sugar and backbones and everything that you need to know. Make sure you know the differences of how they bind together, right? You don't have to know the exact bonding locations, but know that adenine binds with thymine, right? And guanine binds with cytosine. And this is for DNA right here that we're looking at. Very similar to RNA, right, when we look at this one, but you wouldn't have thymine anymore. Yeah, you will have uracil in the place of that binds to these things. So when you look at this one, you want to make sure that you understand the basic structure of DNA, right? It is a double helix. They go in opposite directions, and oftentimes when you see these molecules, the bases are on the inside, and you get the backbone that goes all the way around it, making up the double helices. Okay? Again, just know the basic structure of this and not necessarily have to go into really big detail. Again, if you're taking intermediate cell and molecular biology, then I'm sure you're going to have to be able to draw all of these different types of actual molecules. But for this class, you just want to be able to recognize them, right? Like what is a purine versus a pyrimidine, and realize A's and G's, or guanines and adenines, make up the purines, and the other ones make up the pyrimidines, right, about the nitrogenous bases. So these are the 5 bases found in nucleic acids. We only have 4 per DNA and 4 per RNA. You just want to make sure you know the difference between the two. Okay. Again, a very simple way to memorize which ones belong to purines. I always came up with this idea that pure as silver. If you look at the periodic table, you know silver is Ag, and then you know that pure Ag or pure as silver would be adenine and guanine. Okay. Don't get mixed up this as pure as gold, because then you'll get something completely different, and that's really going to mess you up. Again, this isn't the best analogy, but definitely one to put on the books if you can't think of anything to get that purines versus pyrimidines stuck in your head. Okay, so now when we get into this is the main gist of it, looking at the microbial genetics part of it in chapter 8. Okay? You have to understand how the DNA is made up of these prokaryotic organisms. It's oftentimes just a single circular chromosome. Okay? It's not linear like what you find in eukaryotic organisms, and oftentimes you only have one. Okay? It's a pretty big piece of DNA. You can notice if you unravel the whole entire thing, it's about 1 millimeter. Most cells are only 1 micrometer, so these things have to figure out a way to get it all jam-packed into the one little tiny cell. So there's no histones, there's no proteins involved in the DNA twisting. Oftentimes this is just by supercoiling. Okay? The supercoiling mechanism, if you can imagine it, if you take a rubber band and you keep twisting it and twisting it and twisting it from both ends, eventually it's going to start folding in on each other and get to a very, very tight ball. Okay? Exactly the same thing is happening with these prokaryotic organisms. The amazing part of this is that it still has to replicate, even if it is supercoiled, before it actually does the cell division. So you can imagine if cell division takes 20 minutes for E. coli, the chromosomes have to double in that same amount of time, if not even faster than 20 minutes before the cell splits. Okay, so when you look at this, again, you want to make sure that you understand the differences between DNA replication versus DNA transcription and DNA and RNA translation. Okay, so we'll talk about each one of these things as well as the recombination expression and the replication of the cells. Right now we're going to really work on that cell replication part of it and definitely get into the recombination expression a little bit later. So some definitions, right? Some basic ideas of how these things are put together, right? A gene, we always talk about these things, and this is a basic unit of heredity. It's an ordered sequence of nucleotides located in a particular position on that chromosome, so in that location is called a locus, and that's going to encode for a specific functional product. So if we know what the gene is, or we know what the function of that part of the chromosome is, we call it a gene, right? And that position within that chromosome is definitely a locus. Now, an allele, we don't really have too much of this when we talk into perspectives of prokaryotic organisms, because oftentimes we talk about different alleles. So if you have two chromosomes, you can have two alleles of the same gene, like a dominant and recessive. Prokaryotes don't have this. So the only time we see these alleles is associated with mutations. So these would be genes with different information at the same locus. So maybe you have one gene that's completely changed or have some type of genetic mutation associated with it. Then we start using those words allele. So most often we don't really use it in the same form as we would for those eukaryotic organisms. Okay, so this is just a diagram showing how much DNA is associated with one cell or one E. coli cell. You can see how much more DNA is there that has to jam pack itself into that one cell. And then when you look at an actual cartoon image of it, here is what a circular chromosome might look like. And so basically what they're saying is that everything that has these sort of color coordinated purple or blues or reds, those oftentimes have a very similar function. Okay, so the purples in this case represent DNA replication and repair. And we see the darker blues, which is going to be amino acid metabolism, correct? And the actual red ones are going to be carbohydrate metabolism. So you can see that these are oftentimes spread around the chromosome, but they oftentimes will have very specific locations, right? Like a locus, where you have membrane synthesis. So oftentimes in these microbial systems, you're going to have it very well organized. They're not going to be randomly placed within the chromosome. These have very specific areas that they're being synthesized from, so they can ever be done very, very, very efficiently. Okay, so here's your chromosome, right? And then in this one particular area, not necessarily just that one place, but any one of these you're talking about will have a locus. And within that locus, you're going to have multiple genes encoding for a specific function. In this case, you're having carbohydrate metabolism. Most times when you talk about these things with prokaryotic organisms, you have one gene that's going to produce that one enzyme. Okay? You're not going to have a segment of a gene that's going to produce 3 or 4 different types of enzymes. So it's very simple. It's a one-to-one relationship. One gene creates one enzyme or one protein. Okay, with mutations for prokaryotic organisms means that we're talking about a permanent alteration in the DNA. Because oftentimes we only have one gene, if it gets mutated, we get a permanent change within that DNA. So these change in nucleotide sequences in the DNA can really change the way the DNA is going to be transcribed, which then can be translated. And we oftentimes can get real serious changes associated with these microorganisms because they only have that one gene per one enzyme. Okay? Maybe not so much where you would get major different phenotypes like what you would see here, okay, associated with this little cat, but definitely things that are going to be drastically beneficial or drastically negative for these cells. Okay, oftentimes most prokaryotic cells that have major change to their DNA are not going to live. Right, so when we take about mutations, we oftentimes think of Drosophila, right, and having all these different phenotypes. Yeah, or really crazy phenotypes where you'd have these guys with multiple eyes, right, or you think about getting enhanced type of actual behaviors or characteristics that make you superhuman in such a case. Okay, but oftentimes these genetic mutations you never see. There's no real big genetic changes that are going to be really big changes to the phenotype of these prokaryotic cells. Oftentimes it's very tiny. Structural changes to the protein, right? Maybe you get a better active site associated with the enzymes, or maybe a different structure associated with the membranes. But oftentimes, if there are big changes to the cells, they're not going to be necessarily beneficial, and again, these cells will die. But here's just an example of one gene that can change and can drastically change in one animal. So this is a whippet. Okay, Wendy the whippet has double muscle. Through traditional breeding, oftentimes whippets, they want to be very, very fast, right? So they breed them for having a certain muscle type. But oftentimes it goes a little haywire when they get double muscle, okay, a mutation where they're going to get multiple amounts of actual muscles in their hind legs and their front legs, and they become almost look like the dogs on steroids. Okay, in fact, this dog is not on steroids, not on anything. It's just a normal mutation that happens through traditional breeding. So the DNA stores all the information, transfers information, right? So nucleic acids store all the information for what the cell needs to do, and within those 4 nitrogenous bases, A, G, T, and C. Now it's going to transfer that information into structure or proteins through replication, transcription, translation, right? So we go from DNA to RNA to protein. Now this is the core of genetics when we talk about DNA to RNA to protein. And the reason why I'm saying this right now is that oftentimes this is what they teach you in every single biology class. The reason why you want to pay attention to this one-way street of DNA to RNA to protein is that when we get into viruses, it's going to go all haywire. It's going to change around. Okay, so it's very strict to prokaryotic organisms and eukaryotic organisms because viruses are a completely different type of actual pathogen. It does something completely different. They don't fall within the same category of the things that we found on the tree of life. So they don't respond to the exact same core of genetics. Okay, so everything we're studying here, make sure you understand well, because viruses will throw you for a big loop when we start looking at the replication process of them. You'll notice that a lot of times when replication process happens, okay, we see that the actual bases are inside and we see anti-parallel systems between the actual backbone of the DNA. The major thing you want to realize is that replication oftentimes has a 5' end and a 3' end for DNA. So all times you have the DNA added to the bases, or the bases added to the DNA, it's always going to happen to the 3' end. Okay, you're never going to have it add to this end. It's always going to happen to this end of the actual growing strand, or here. Okay, so it's a very, very specific area where you can add nucleic acids. Nucleotides too. So we see a lot of this process happen, and we know it base pairs to A's to T's to G's and C's that happen over and over again through the replication process. Okay. Again, very, very simple things that you should have learned in your basic biology class in high school, or maybe another class that you've taken elsewhere. Okay, but you want to know the basic things that are happening with DNA replication. Yeah, you have to realize that with DNA replication, the strands have to split, right? And you have to add growing strands on both ends of it. So when we see these type of prokaryotic systems, oftentimes we talk about the replication fork. Okay, so if you see here a double stranded of DNA, it will split into single strands. Okay, and then you oftentimes have replication on one strand, and you have replication on the other. Both will abide by the same rules by adding A's to T's and G's to C's by matching it of whatever the parent strand is, the new growing strand. But you're going to have the same enzymes that are going to come into play to do this actual work. And so you want to make sure you understand what's happening on each one of these replicating strands. For example, if you take this cartoon image of what's happening with DNA replication, you got to imagine that this actual area within the cell, we have a circle, right? And within that circle, you have to have the DNA splitting at a very specific site. And that specific site is called the origin of replication, and this is where it initiates the actual replication process to begin to split, okay, or a bubble to happen. So the double strands begin to separate into single strands. And when single strands happen, you're going to have 2 replication forks occur, right, at the one of each end of the bubble. Meaning if we have it right here, you're going to have 1 replication fork there and 1 replication fork there. Okay, so 2 replication forks every time you have DNA replication. So in this case, you can see how fast replication can occur. You don't have to replicate all the way around the whole entire cell at one time. With this mechanism, you only have to do half, right? And this one is going to go the other way, and then they will meet and they'll fall apart. What's happening in each one of these replication forks is what's being shown here in this figure. you can see multiple different parts that are happening within it, but you can see the major players being very similar. The easiest thing to think about is that when it splits, you're going to have major tension associated with this actual section. So oftentimes you're going to have enzymes that are going to help release that tension as long as breaking the strands. You need proteins involved in stabilizing these single strands because they have complementary base pairs. You don't want them to stick back together again, so they help stabilize that single strands. And then from there, you're going to have the growing strand of the DNA through DNA polymerase. Okay? You don't have to memorize a specific type of DNA polymerase. You just want to know that this actual enzyme is a polymerase that converts DNA to DNA is called a DNA polymerase. Okay? And from this, it grows on from the growing strand. So here they don't really show this, but oftentimes there is a primer involved in this, a very short piece of RNA that gets added onto it that allows for the DNA polymerase to add on the complementary bases. And by doing so, you can see that it's a continuous strand because on this one, it's adding it on the 3' end, and it can keep doing it over and over and over and over again. Okay, we call this the leading strand. Because the other strand is the opposite orientation, it has to be put together in parts. Right? Remember the rule of always adding on to the 3' end. In this case, the 3' end is always going to happen on the opposite one. So it's going to be growing the strand in the opposite way, whichever way the actual replication fork is opening. So in this case, every time the RNA primer gets placed down through a primase, we can see that the DNA polymerase is going to grow it in the opposite direction of the way the replication fork is opening. Because it's doing these in sections in this way, right, and it has to do it in small fragments, we call this a lagging strand. Okay? The lagging strand is also known as Okazaki fragments because of the short sequences that it gets produced, but it gets all put back together again through a ligase enzyme, which secures everything back together again. So what we're talking about here is that a replication fork. And remember, replication fork is a point in which the two strands of DNA separate for replication. Right? For every chromosome, we have two replication forks if we have that one origin of replication. The DNA polymerase is the enzyme that synthesizes the new complementary DNA. And remember that origin of replication is that single location on the chromosome where the DNA synthesis is initiated. Okazaki fragments are short DNA fragments, right? And the DNA ligase seals any nicks made in DNA synthesis and also assists in the DNA repair. So this helps piece everything back together so you have a nice strand that goes back and you get complete replication. The helicase is that protein that sits in front of the replication fork, and that helps unwind the double-stranded DNA to make single strands. Right? So basically, these are the major terms that I really want you to understand from replication process, and knowing that the complementary base pairs happen between each one of the parent strand and the new strand that's being produced. What we see with this actual type of replication is what they call semi-conservative replication. We talked about a parent strand, and we talked about a new strand or a daughter strand that's being produced. Here we see an origin of replication, right, that's being split and two growing strands being produced. As it keeps moving in one direction and the replication moves forward in the other, you're going to have two of the actual strands being reproduced at the exact same time, okay, from parent ones. So if you can imagine, you have one old strand and one new strand. And we call this semi-conservative replication because we're always going to get a copy of the original, right? So you can imagine you have an original piece of DNA and that gets copied. You never really want to have a copy of a copy which then gets copied again over and over and over again, because if you know by using copy machines, you kind of lose any information that's associated with that paper if it's a copy of a copy, and which then makes another copy of it. So by always keeping the original there, you're always going to get the information restored to that new strand of DNA. Now moving into the transcription of RNA, it's very, very similar to DNA replication, but we're converting DNA to RNA. And because we're converting DNA to RNA, we call this an RNA polymerase. And because we don't have any thymine associated with RNA, we're adding uracil into this, right? So that's going to be in the place of any thymines that are doing this. So you can imagine that we have a very specific area of transcription, and we'll get into all of these very specific areas like the promoters. But here's your major enzyme that's doing an RNA polymerase. It's going to add the actual bases to the complementary strand of DNA. So it's a single strand of DNA, right? Just like how you would have for replication, but instead of growing a DNA strand, you're growing RNA to make it actual use for later on for translation. So as the bases add, you'll have the same C to G complementary bases, but you can notice here, you're not going to put a thymine in the place of the A to T. You're going to have uracils, because uracil is only found in RNA. As it reaches the end of the sequence before it finishes off that transcription, it breaks off and you're left with your RNA. Okay? So in this case, your RNA is always going to be a single strand of RNA. You do not have double-stranded RNA molecules within these type of prokaryotic cells. It's very, very different if you have these things, and you're going to find out the reason for it when we start looking at the viruses. But basically, the RNA polymerase begins, and it ends at a very specific termination sequence, thereby producing that RNA strand that has uracils instead of thymines. So when we look at this in a very short description of it, the transcription begins with the RNA polymerase binds to a very specific sequence of DNA. We call this the promoter sequence. Okay, this helps open up that DNA double helix, and it's going to assemble or produce the new type of actual RNA by putting in the correct bases. The ribonucleotide is inserted into the growing RNA strand, and again, it follows the same base pair rules, right? G's to C's and A's to U's this time. And again, it happens in the exact same process as DNA replication, right? We still have the 5' to 3' growing strand, but the only way that it finishes is going to happen in a termination sequence. Okay, so a little difference is associated with this in replication, DNA replication, but you can imagine you have very similarities between putting the base pairs there. How do you initiate DNA replication? It's an origin of replication. How does it terminate? It terminates when the two strands come together. When you talk about initiation of RNA transcription, the way it begins is having a promoter sequence. The way it ends is having a termination sequence. So from RNA transcription, we have three types of RNA produced. You have mRNA, rRNA, and tRNA. Messenger RNA is going to be translated into that polypeptide. Okay, that's the information that has everything there. That's the blueprint of the RNA. Ribosomal RNA is a ribosome, right? This is going to be used to build the ribosome's machinery synthesized proteins by translating the mRNA, right? You now know that ribosomes produce proteins. Well, here's the RNA that does it. Transfer RNA are these special RNA molecules that carry the amino acids to the ribosome to build off of that messenger RNA to produce that polypeptide or those proteins. Translation requires all three of these things. Most times people think only messenger RNA produces the protein. Well, in fact, you need all three types of RNA to produce that protein. Okay, so let's go back to mutations really quickly and show how we can have differences associated with this transcription to produce these different types of mutations, right? Now, mutations are changes within the genetic material, right? You can have neutral mutants, you get beneficial or harmful ones. Most times when we talk about spontaneous mutations, it's 1 in 1 million base pairs. So 1 in a million, we have that rate of a spontaneous mutation, which means every time you have replication of that chromosome, you probably have one mistake that might be happening within it. But if you apply a chemical or you apply something that causes mutation rates to increase, like a mutagen, you can have 10 to 1,000 times more frequent. Now, having these type of changes, we oftentimes have these things called base substitutions or point mutations. And these oftentimes change in one base, meaning you can have one base change that can cause drastic differences associated with it. So in this case here, we see a change of a G, a normal one having a C, C, G, G here, if you see here. But in this case, you had it change to a T, and that's going to, again, of course, change that complementary base pair because it's matching it off of that sequence. From this type of actual information, we can change the amino acid sequence of the polypeptides being produced right through this missense mutation. Instead of getting a glycine, we're going to get a serine through the translational process, which can drastically change that type of protein, or not. Yeah, it depends on how the actual mutation really occurs or not. But in this case, you can imagine just having one base change can drastically change this type of actual peptide. Nonsense mutations are really detrimental to the cell. Most times you're going to kill the cell because you can imagine nonsense mutations produce nonsense codons or stop codons, right? It basically eliminates the whole entire protein. So in this case, if you have a drastic point mutation or a base substitution very early on in that translational process or building that RNA molecule, you can accidentally place a stop codon early on, causing this nonsense mutation. Frame shift mutations are very similar. This is oftentimes if you insert a piece of DNA or you delete a nucleotide, you can drastically change that sequence as well. So you can imagine just having very, very, very small changes within the DNA can lead to big, big changes for these prokaryotic organisms. Okay, so when we look at these, we can have chemical mutagens that can do this. For example, by adding certain types of chemicals to this, we can actually change the A's to bind to C's. Yeah, and oftentimes you don't want to have this, but you can do this to make these type of mutations occur. Radiation can definitely do this, right? If you apply a lot of UV light, oftentimes you can create thymine dimers. Okay, thymine dimers cause it to bind together, and if it binds together, they cannot bind to the complementary A's, and so you're basically eliminating that section, and you're causing major disruption into this actual DNA. So this is how UV light causes major problems associated with the DNA. Okay? Oftentimes we do have a system that deletes it and you can produce new ones to fix it, but you can see how quickly UV radiation can just cause this thymine dimers to occur and can really mess up the whole entire DNA. Now, we use this type of mutations very uniquely within the health field, and I wanted to spend a little bit of time describing what the Ames test does because it's an important assay to look for carcinogens. So carcinogens, you now know, are things that causes cancer, right? In humans, in people, in eukaryotic organisms. You can imagine having a way to screen these type of carcinogens to see what the mutation rate might be or how they cause cancer would be really helpful to us to screen a lot of products or chemical compounds that might be used on a daily basis. Okay, a lot of the carcinogens that we found today are developed through this Ames test. So the Ames test uses a mutant bacteria which are going to be exposed to these substances or carcinogens which cause the mutants to revert back to the original phenotype. Okay, so it's a little different. You might think of it, oh, how does it increase the mutation rate of these actual normal wild types or the original phenotypes? But actually we're looking at the reverse of it. We have a very specific mutant and we look at how that chemical converts it back to the original wild type. And the reason for that is that oftentimes the high rate of mutations within these prokaryotic organisms is going to block the effects of this actual carcinogen. So we look for a very, very specific compound that can really revert back that mutant into the original phenotype. And we know that probably causes a lot of change to the DNA, which is going to mean much more carcinogenic compound. So what we're doing here is oftentimes we'll look at a control substance, right? Which means if you have mutant bacteria here, okay, and your mutant bacteria cannot grow on histidine, okay, a nutrient that's there and it's lacking in there, right? So it can't produce it, so it's going to be not be able to grow on it. So if you put your compound in there, right, and it doesn't do anything, if you plate that bacteria, it's not going to grow, right? It's a mutant of it. It's not supposed to grow. So after growing there, you might find a few cells due through normal mutation rates that happen because it's 1 in a million, right? Because you have so many cells and bacterial cells associated, you probably will get 1 or 2 that can grow on top of that media, which means you've changed that DNA to convert back to the original wild type form or the original phenotype. Now, here's your background information, right? So you do get a few cells, but not that many. Now, if you put in your experimental one with the suspected mutagen or that chemical compound that might cause cancers or carcinogenic compounds, and you mix it with your rat liver extract, right, with that mutant bacteria that are not supposed to grow on the media lacking histidine, and you incubate it, and all of a sudden, all of the cells are now able to revert back to the original phenotype, you now know that this compound causes major changes to the DNA that really cause these guys to mutate back to the wild type. So you know this is a really strong mutagen or carcinogenic compound that can really affect the DNA. Okay? A lot of times these compounds can do so, but if you look at this very specifically, we're adding rat liver extract. Okay? Why do we add rat liver extract? Well, through metabolism, okay, oftentimes the compound that's being ingested by people are not actually carcinogenic. It's the breakdown products. Okay, so if you ingest something, oftentimes that compound might not be carcinogenic, but its breakdown products are. And that's what the liver extract is doing, is breaking it down into smaller compounds that might be the actual compounds that cause cancer or carcinogenic compounds. So the question of the day, right, the last thing that I want to stop with for this lecture is that the nucleoside analogs and ionizing radiation are used in treatment of cancer. Right, we now know this. You can give these type of actual compounds, right, that causes major damage to the DNA. So why do you suppose they're used to treat some kind of disease, even though you know they cause so much damage to the DNA?